originally from Waco, Nebraska, and this is my presentation. And I was really curious about like P53 and telomerase. So I was trying to figure out the correlation between the two proteins and enzymes. And this is what we're going to discuss. And it's the protein and the enzyme, plus my personal research, and also past research, and also other animal models that possibly have these same genes. And I'm going to talk about why I chose this topic. I was originally studying the AIRE gene, the A-I-R-E gene, and the correlation with P53. And with that gene, is found inside the thymus cells, which is and it activates other proteins to be created inside cells that normally are turned off. And what I found was there was no really correlation between the two proteins, so I decided to switch to telomerase because I found a news article talking about bats. It turns out that bats do not develop cancer at all. There's like barely any reported cases, so we looked into it, and they found that bats have 2 copies of telomerase enzymes, and they have 2 copies of P53, which are genes that we also have, but somehow they do not end up with cancer, which sparked my interest in how this was related to us. And I was also doing cell culture, so I decided to use some of the same cells and start researching them. And some background research about what some of these genes kind of do. I'm going to talk about the central dogma of DNA. And what happens is that our DNA creates the mRNA, then the peptides. And I'm not going to open up the can of worms to how that had actually changed. And anytime that you hear the word ACE at the end of a protein, it means that it's simply an enzyme. And we're going to talk about some of the cell cycles. And some proteins are multifunctional, meaning that they have more than one function that can influence other proteins. And these are the 4 peptides, that peptide nucleotides, that make up DNA. So all of our DNA genome comes down to these 4 little letters. And these will come up again when I talk about telomeres. And some genes are also promoters, which means that help turn on other genes. We're going to talk about P53, also called the guardian of the genome. P53 is mutated in 50% of cancers. So what does P53 do? P53 helps monitor the health of DNA. Mutations can lead to cancer, and this is because when your DNA becomes mutated, you have an increased risk of genes that influence cell cycle receptors, and cell cycle arrest are then influenced, because cancer is simply where your cells are not being told not to divide. It's simply uncontrolled cellular division. So P53 has different functions depending on the amount of protein that's found within these cells. And so P53 triggers cell arrest at the G1 phase and the S phase. And the S phase that you see up there is about DNA synthesis, and you would like for your cells to not start replicating your DNA if it's highly mutated. We also have a stop point at G2/N phase, or not N phase, but the checkpoints, to prevent DNA damage, just in case the cell starts to divide if there is DNA damage. The moderate level of P53, we find that it starts turning on other genes, like P21, NOVA and PUMA, and WIP1, and these help stop the cell at the S phase to prevent prevent the cell from going past any more checkpoints. And this is probably one of the most important functions of P53, and it's apoptosis. Apoptosis is programmed cell death. So if a cell has high amount of P53 and large amount of DNA damage, it has 2 options. Either it goes to synapsis, where it goes to sleep, or it has to go through cellular death. So we would rather have our cells, simply one of them die off, or essentially kill themselves, rather than having them turn into cancerous cells. So what our cells do is that P53 is activated by DNA damage, which leads to, let me pull it up. It shows right here, BAX. It activates BAX, which then interferes with the mitochondria, which leads to capsaicin, which leads to apoptosis. The capsaicin protein will start to interfere with the cytoskeleton, causing the cell to fold in on itself. Essentially, it makes the cell become a black hole, and it dies, and macrophages, which are part of your immune system, come along and eat up the cellular debris like nothing happened. And we're talking about telomerase. And sometimes it's considered a semi-proto-oncogene. What this means is that proto means first, and onco refers to tumor, and gene refers to genetics and heredity. So it's a pro-oncogene, the gene that influences the cell cycle and tells the cell to keep dividing, while also increasing the risk of cancer. So cells that are cancerous typically will have a mutated form of telomerase. So when we're actually testing for cancer, sometimes we will also look for telomerase expression. So what is telomerase? So telomerase essentially lengthens the telomeres. The telomeres are the end of our chromosomes, and they will slowly over time will be destroyed or lost because of how our cells replicate our DNA. And so essentially the telomeres are repeating parts of DNA. There are T, A, G. These are they were before considered junk DNA. Now they actually have a function of protecting the rest of the genome. So at the end of the chromosome, you have these little T loops up here. These are kind of like the shoestring caps of our chromosomes, help keep our DNA from unraveling and prevent it from falling apart, because if the telomeres are shortened, it starts to eat away at our actual genes that we actually need. Another major function of our telomeres is that it also signals to tell the cell to stop dividing. When the cell keeps dividing while it has shortening telomeres, it gets to a point where they start to eat away at the genome. That becomes a major problem because it also reflects the age of the chromosomes. Your cells can only divide upwards of 20 to 50 times before they have to shut down, because that also reflects the age of the DNA. If you guys are familiar with the game of telephone, that's kind of how our DNA works. From one cell to the next cell it creates, which leads to over time the gene may change and mutations may happen to where they no longer would be safe to keep dividing. And telomerases will help extend the ends of the chromosome, because the DNA polymerase, is unable to get to the end of the DNA because it can only work from a 5' to 3' end, so the lagging end will have to be shorter. So up there we have reverse transcriptase, which is what I also researched too. That's an essential part of the telomerase. So it allows the cells to keep lengthening the telomeres, which is a signal telling the cells that, oh, we're still really young, which they aren't. Which is also what happens inside of stem cells. They have active telomerase, and cancer cells also have active telomerase, which is how we have immortal cell lines. In personal research, I was researching with HeLa cells and CaCO2 cells, or CaCO cells. Those are adherent cells. What that means is that they stick to the bottom of the flask when they're exposed to media. And with those cells, the HeLa cells are cancerous cervical cells, and the CaCO2 cells are cancerous colorectal cells. Then we have RPMI cells, the 8226 cells, and those are non-adherent. But what that means is that they're suspended aside the media, to where if you were to dump out your media with those cells, you would lose all of your cells. And with that, you would have to have two different types of media. We have RPMI media, and we have EM media. EM is minimum essential media. And then we add 10% fetal bovine serum and antibiotics to it. So what I chose to do was a Western blot. And what this is where we are looking at the expression of proteins. I chose to do a Western blot over doing a Southern blot or Northern blot, because a Northern blot shows if a gene is there. A Southern blot simply shows that RNA is there. But a Western blot, if you remember the central dogma of DNA, all proteins are made from RNA and then from your genes. So what we do is we have to prep our sample. So what we do is spin out our cells and we wash them with a buffer, and then we have to heat them up. We have to lyse them, then we have to get the proteins out, then we heat up the sample with our loading dyes so we can easily put them inside of our gel. And we pipette into a polyacrylamide gel, which is kind of a textured jello. And it is ran through the gel, and the protein will move through the gel because proteins are negatively charged. So it's actually an electrical current that runs through there. So when it's exposed to electrical current, the protein will move to the positive side, moving away from the negative part of the gel, which will create bands because the larger proteins move slower, so they stick at the top, and the smaller ones move through the gel to the bottom. And what we have to do next then, because it's like a jello texture, we have to use a blotting paper with it. Not a blotting paper, but a cellulose membrane. So what we do is we take it's kind of like a machine, like a square machine, and it's the same type of idea where the bottom part of it is negatively charged, and the top part is positively charged. We run an electrical current through it. We take blotting paper, and we have the membrane soaked with a buffer. They put our gel, then we put our cellulose membrane. Then we put another piece of blotting paper, and we squeeze together. When the charged electrical currents run through it, the proteins move from the gel to the cellulose membrane. Therefore, the cellulose membrane has a reflection of our proteins. So we'll be able to see it, and we're able to actually use it now. And so the next step was to expose to an antibody. But before that, we added a generic protein. Because the cellulose membrane is very adherent to proteins, if we were to just randomly add the antibodies to it, it would have stuck all over the membrane. So what we do is we block it with non-fat dry milk. So if you're seeing anything with labs, it has an image of, if you see non-fat dry milk powder, it's what we're using inside of our membranes. So we block it overnight with the powder inside a buffer. Then we rinse it off. Then we add a primary antibody. And what that is, we use a P53 antibody, and then we use a TERT, what you saw earlier, antibody. And those antibodies are very, very specific, and they'll only bind to those proteins. So we expose the membrane to the primary antibodies. And then we incubate to another antibody to help straighten the signal. Because the antibody is from two different animals, therefore it will have a mutual response, technically, to another antibody and bind to it. And after that, we put a chemoluminescent chemical on it, so that when we image it and UV rays, we'll be able to see all the bands. And so with my research, we attempted this, and we could not get a Western blot to show up with the membranes. So we tried different things, but we still couldn't really get it to work. And so overall, this would have shown if the two proteins were there or if only one was there. And also, we were able to use both the antibodies at the same time because P53 and TERT are drastically different sizes. So I'm going to talk about past research. Some of my research has concluded that if telomerase is active, the length of the telomeres are extended, leading to an increased cellular division capacity. More a cell replicates its DNA, the older the DNA becomes. This leads to more mutations and activation of P53. Telomerase can indirectly influence P53. There was a study that was done with 158 participants. 4% of participants were men. What they were doing was studying the reverse transcriptase and mutated P53 inside bladder cancer. So what they did was they took samples of the tumors from the bladders, then they exposed them to immunohistochemistry analysis, where they did the same thing that you would do with a Western blot, where you expose the samples to antibodies and see what colors they glow, showing if it was there or not. And so overall, we found that there is a supportive evidence to show that functional TERT with a mutated P53 increased cancer growth. And if a protein is mutated, it may not function. So it's actually a broken protein. And there is an association between mutated TERT and tumors with a high amount of protein 53. We also found some unsupportive evidence that if both genes are mutated, there's like no correlation between the two. And there's also no association between a mutated telomerase reverse transcriptase and a low level of P53. And how they actually tested this was that when they exposed the antibodies to the UV lights, they would have a scale and like for the staining. And so they had a scale from like 0, 1 to 10, 11% to like 50%, and then 50% to 100%, which was like very odd because like it was like eyeballing kind of when they did a study. Okay, we're going to talk about human papillomavirus. Another study was done with this cancer-causing virus. This virus is able to infect our cervical cells and other cells in our bodies. And they normally infect our keratinocyte cells. So what's the major part about this virus is that it's a DNA-based virus. And DNA-based viruses are able to incorporate sometimes their DNA into our DNA, which then causes some of our cancer problems. And not only that, sometimes they evolve other proteins that interact with our own proteins and degrade them. And so this may not make sense, but I can explain it. With infected cells, there's less telomerase but more P53. If they're cancer-causing cells, why are they decreasing some of our cancer-causing protein enzymes and increasing some of our tumor suppressors? It's actually to slow down our cell cycle so that the virus is able to put itself inside the DNA. It'll be very, very hard to if the DNA is being replicated. And then also it will degrade protein 53 by using their own enzymes, E6 and E7, and it's done by adding a ubiquitin tag to P53. And ubiquitination is where a phosphate protein is added to when ubiquitin is added to a protein, which leads it to be exposed to a protein that will end up breaking it down in complex. This is also how some of the cells some cell lines also are immortalized with P53. And from another study talking about birth control and Cahat, you know the image earlier, I'm going to go back. This is a plant that is from the Eastern Africa, as what else? The other peninsula, where you go chew it, kind of like chewing tobacco. And it has the same type of effect as epinephrines, and it's also banned in certain Western countries because of health risk and addiction risk. We did a study comparing these two with women who have breast cancer. So the 140 women that were a part of this study, 60 of them had breast cancer, and so the women were placed into separate groups. Well, if they were on birth control for the past year, they were placed in one group. In the past year, if they had been chewing Cahat for the last year too, put it to another group. And the birth control they were on had both progesterone and estrogen. And not only that, breast cancer patients were not separated from these groups. So breast cancer patients were in both groups. And they did a study where they compared, let's say a patient had breast cancer, Cahat, and also had birth control. Or one person was written down that, hey, they were on birth control but did not have breast cancer, but also Cahat. So what we found was the telomerase, that all women who took birth control, they all had increased telomerase. So that could be linked as to why sometimes some birth controls increase the risk of cancer. We also found that 27.2% higher in women with birth control users than non-birth control users. And that women without cancer had high telomerase and low P53, which makes sense if your cells are working, not doing, you shouldn't really have high amount of P53. And then of course, P53 was significantly higher in breast cancer patients that were on birth control. And we didn't really see a really big difference with telomerase, but we found that 39.6% increase in P53 in non-breast cancer patients. So we would have compared, let's say, a patient who was chewing Cahat but not on birth control, who had breast cancer, to another patient who was chewing Cahat and was not on birth control. So they compared patients to the same patients and back and forth. So the next one was like 27.3% increase in P53 compared to not Cahat chewers overall. And we're going to talk about animals. It's really surprising how many animals throughout the animal kingdom are way more susceptible to cancer, but the other ones are more susceptible to cancer. And there's an animal that we normally use a lot in lab, other than mice. We can use zebrafish because they're quite easy to maintain. So in this research, they were researching the fertility of the male zebrafish in relation to P53 and telomerase. So in this study, they found that they took male zebrafish, mutated the P53 gene and telomerase. The male fish that had both the P53 alleles mutated, had a drastic decline in cell division and proliferation inside the testes. A lot of them became sterile, most of them did. And that zebrafish with mutated TERT started having severe digestive problems, where the cells inside the digestive linings were not dividing. And some of these male fish, they then crossed them over and crossbred them with unmutated female fish. Then the number of eggs were assessed. It was found that male zebrafish who had both their TERT genes were mutated by the age of 6 months. They were completely infertile. And also, zebrafish have the same length of telomeres as humans. Again, like humans, zebrafish who had P53 mutated, it led to them having soft tissue tumors. And both of those proteins and enzymes drastically impacted the male's fertility. We're going to talk about elephants. Elephants have 19 retrograde copies of P53. Retrograde means that the gene lacks introns. Introns and extrons are parts of RNA where they are left inside of the nucleus, and the exons exit the nucleus to form the mRNA. And so these are copies of the gene that were re-put into the genome. This sometimes happens from retroviruses. So elephants have been around for 60 million years. And elephants are huge. You all know that elephants are huge, which means that they have a lot more cells to take care of. And they live for quite a long time compared to other mammals. Therefore, overall, they have a very high risk of cancer. Therefore, there was high selective pressure to actually use these 19 retrograde copies of P53. Not only that, but elephants live in such a very high temperature area, and their body is so large that they have to have regulators inside there to help maintain their body heat. Because in our normal cells, if we're overexposed to high temperatures, it will start to deteriorate our proteins. But in elephants, they have enzymes that are able to work at higher temperatures, which allows their cells to be protected from stress and oxidation, which decreases the risk of cancer. Not only that, but elephants are strangely related to other killer whales and other large aquatic mammals that do not inherit cancer. That's why there's the image of a killer whale up there, because they don't get cancer either, and elephants and those two are very closely related. And in this study too, they talked about how they're related to hyraxes, which are small little mammals, and they also have P53 genes. And we're going to talk about one of my favorite animals, bats. This was very shocking to talk about how bats, they're a little more closely related to us than other mammals inside the labs. And they live up to 20 to 30 to even 40 years, depending on their species, because this is a very, very broad order of animals. And so bats, they also have menstrual cycles, menopause. They have to have extremely, their ears are very, very important to their hearing compared to ours. Therefore, they have to make sure that survives their whole life. And they also have very, very fragile skeletons, and they're exposed to very deadly viruses. And they also have to take on the roles of like flying because it's very hard on your metabolism. So all of these things increase your risk of inflammation and cancer. So what the bats' bodies did was essentially evolve better P53 and telomerase because it helps with their cells when they're exposed to viruses. And when also exposed to viruses, their immune systems have better interferons, which help eliminate inflammation, because when they're exposed to deadly viruses, it creates a lot of cellular death. Therefore, your body has to find a way of controlling it, which is better than our bodies can, which is why they do not get cancer. And also, during hibernation, it's found that some mammals, including bats, during hibernation, it lengthens their telomeres, while having to have telomerase active. There's also an initiative to actually track down the whole genome of multiple different bat species too. Not only that, because bat skeletons are so fragile, they always have to have their cells replicating. Therefore, they have better telomerase inside of their fibroblast. And some of the conclusions. There is some evidence to support the possible link between P53 and telomerase. And I do want to continue working with some of these cell lines to see if they actually do produce these proteins. Because if a cancer cell, let's say HeLa cells, they're cancerous, which means that they probably have telomerase active. That's what keeps them immortal. But I don't know whether or not they actually have P53 or not, because P53, in some cases, can also be a cancerous protein. And also, we need more funding for misfit animals and bats, because sometimes there's very odd things that we're now finding out today. A lot of this research is very, very recent with bats, because they finally got funding for it. And this is it. And I'd like to thank Dr. Finkelstein. I'd like to thank my family, because these are people that inspire me to keep continuing research. And I like the STEM program too, because it allows me to present my work at NAS, also Wayne State College's Life Science program, and that's really it. And also Shannon too, my friends. Everyone's supported me through the years.